Hi, my name is Aidan, and I'm a postdoc in the Transformational Bioinformatics group at CSIRO. In this video, I'll be talking to you about how we're developing computational safeguard to detect gene drives in wild populations. To give you an idea of what I'll cover in this video, I'll start by talking about genome insertions, where they might come from, and why we might want to know where they came from. And to put things into context, I'll talk about gene drives, what they are, and how they can be used to manage wild populations. I'll then briefly talk about how we can determine if something has been inserted into the genome, some of the challenges that we may face, and discuss what we're doing to address these challenges. Specifically, I'll talk about how we're using these things called genomic signatures to detect gene drives within a genome, and I'll present some preliminary results of our analyses so far. And finally, I'll conclude with a few remarks about our work. For those who may not know, genome sequences can be altered in various ways, and one of the alterations that can occur is insertions. These insertions can be relatively small, such as single or few-based insertions, or relatively large, such as whole gene insertions. The ones we're interested in and I'll focus on are the large insertions. And one of the questions we had regarding these large insertions was, where did the insertion come from? So we know that some viruses can integrate into their host genome, which is a crucial mechanism for their survival. We also know that some bacteria can develop antimicrobial resistance by integrating antibiotic-resistant genes from other bacteria into their genome. But apart from these cases, genome editing technologies have made it possible to engineer and insert things known as gene drives, which allow us to spread desirable traits through a population. So given that insertions can come from various places, the next question we had was, how do we know whether something has been inserted into the genome? And more precisely, can we tell where it has been inserted? So to illustrate why we are interested in this question, I'll focus on gene drives and how they can be used to manage wild populations. So what are gene drives? Gene drives are genetic elements that have a higher chance of being inherited. They can do this by cleaving the corresponding locus of chromosomes lacking the gene drive, which leads to copying of the gene drive onto the damaged chromosome, and therefore an increased likelihood of being inherited. Because gene drives have a high chance of being inherited, they can spread through a population over many generations, even if they reduce the fitness of individuals. That being said, if we were to release genetically modified individuals containing the gene drive into the wild, we would have the potential to spread desirable traits through a population, which enabled us to control wild population. Because of this, gene drives have the potential to address some key health, environmental, and ecological issues regarding invasive species, disease vectors, and endangered species. So coming back to why we're interested in determining whether something has been inserted into the genome. We are interested in this because it could help us to improve the traceability of gene drives and therefore reduce the risk associated with releasing genetically modified organisms into the wild. But besides gene drives, determining whether something has been inserted into the genome could also help us to improve the detection of viruses that integrate into the genome and cause disease, thereby reducing the risk associated with viral vectors that are used for gene therapy. Furthermore, it could also help us to improve the traceability of antibiotic-resistant genes and bacteria, which would help us to inform treatment options against infection. But how can we know whether something has been inserted into the genome? So if we know that something has been inserted and we know what we're looking for, then we can simply compare a query sequence against a reference. So for example, we can identify regions that are different by comparing a query sequence against a reference. Or we can identify the inserted region by comparing a query sequence against a known inserted sequence. But what if we don't know what we're looking at, or we don't know what we're looking for? So in other words, what if we don't know anything about the host sequence or the inserted sequence, and hence we have no reference sequence or no known inserted sequence to compare against? Also, we don't know the location of the insertion. So to address this problem, we're developing a method based on approaches used in metagenomic analyses. The method involves analyzing the characteristic frequency of oligonucleotide sequences, which we refer to as a genomic signature. So to do this, We construct a frequency vector for each sequence by finding all possible subsequences of a given length and count the frequency of each subsequence. By analyzing the frequency vector of each sequence, we can determine their genetic similarity. Specifically, we would expect that closely related sequences will produce frequency vectors that are similar, and that distantly related sequences will produce frequency vectors that are different. So to demonstrate that closely related sequences have genomic signatures that are similar, and that distantly related sequences have genomic signatures that are different, we first analyzed the genomic signatures of different eukaryotic genomes, namely yeast, Drosophila, zebrafish, mouse, and human. And so for each genome, we calculated their genomic signature. We also randomly sampled 100 10,000 base pair regions from each genome and calculated the genomic signature of each region. And finally, we analyzed all the genomic signatures using TSNE and plotted the first two components to visualize the results. So the plot here shows the genomic signatures of each eukaryotic genome, represented by the squares, and of each 10,000 base pair region, represented by the circles. From the plot, we can see that the human genome in red is positioned closer to the mouse genome in blue, compared to the Drosophila, yeast, and zebrafish genomes in purple, black, and green, respectively. This indicates that the human and mouse genomes are more closely related than the Drosophila, yeast, and zebrafish genomes, based on their signature. We can also see that the 10,000 base pair regions are positioned relatively close to their respective genomes. So for example, the 10,000 base pair regions from yeast are relatively close to the whole yeast genome. Interestingly, however, we see that the 10,000 base pair regions from mouse and human partially overlap, which indicates that some sequences will be difficult to distinguish based on their genomic signatures. Nevertheless, our results demonstrate that different genomes and their subsequences can display unique genetic signatures, and that this can be used to differentiate DNA originating from different species. So given that different genomes display unique genetic signatures, we then investigated whether we can use this to detect gene drives that have been inserted into the genome. To do this, we simulated the insertion of a gene drive into a genome by constructing a theoretical sequence. To construct our theoretical sequence, we randomly inserted the Cas9 gene sequence into a yeast chromosome sequence, and we did this in yeast due to its relatively small sequence. We then used the sliding window approach to calculate the genomic signature of 10,000 base pair regions at different positions along the length of the theoretical sequence and of the native yeast sequence. Finally, we analyzed all of the genomic signatures using TSNE and plotted the first two components to visualize the results. So the plot here shows the genomic signatures of each 10,000 base pair region at different positions along the length of the theoretical sequence and the native sequence. So the signatures from our theoretical sequence are highlighted in blue, and the signatures from our native sequence are highlighted in red. And from the plot, we see almost no blue points. However, upon further inspection, we find that almost all the genomic signatures from the theoretical sequence overlap with the genomic signatures from the native sequence, which was expected since almost all of the sequences for each region in the theoretical and the native sequence are identical. Interestingly, there were some genomic signatures from the theoretical sequence that did not overlap with the genomic signatures from the native sequence and hence are visible. Specifically, the points around the top left corner all correspond to regions that contain the Cas9 sequence. So in summary, our results demonstrate that the gene drive and the native DNA sequence can be distinguished based on their genomic signature. And this highlights that it is possible to detect gene drives that have been inserted into the genome. So in the previous analyses, we focused on whole genome sequences. But what if the genomes that we're interested in are incomplete, fragmented, or newly sequenced? Are we still able to detect whether something has been inserted using our genomic signatures? To investigate this, we generated an Illumina sequencing library from our theoretical sequence. We then calculated the genomic signature of each sequenced read, analyzed all of the genomic signatures using TSNE, and plotted the first two components to visualize the results. So the plot shows the genomic signatures of each sequenced read. Genomic signatures of reads mapping to the Cas9 sequence are highlighted in blue, while genomic signatures of reads mapping to the rest of the theoretical sequence are highlighted in red. And from the plot, we can see the relative similarity between reads based on their genomic signature. We see that some of the reads are positioned relatively close, indicating that their sequences are similar. And because of this, we can start to see a few groups of closely related sequences. We can also see some of the reads mapping to the Cas9 sequence are closely related. However, we see some of the reads mapping to the Cas9 sequence are also relatively different. And because of this, they are spread out across the whole plot. And this raises the question of how do we actually determine which reads map to the Cas9 sequence and which reads map to the rest of the theoretical sequence. So from our previous analyses, we reasoned that the genomic signatures for each sequenced read could serve as an estimate of what the true genomic signatures might be. That means we can get a better estimate of what the true genomic signatures might be by calculating the average genomic signature of sequenced reads that are similar. Specifically, we can construct frequency vectors for each closely related sequence and take the average of these frequency vectors, which would result in a frequency vector that corresponds to the average genomic signature of a sequence. We also reasoned that since the length of the inserted sequence is likely to be relatively small compared to the length of the native sequence, that we would expect that the average genomic signature of all sequence reads and the average signature of sequence reads mapping to the native sequence will be similar. By contrast, we would expect the average signatures of all sequenced reads and the average signature of the inserted sequence will be different. Therefore, if we can compare the average genomic signatures for groups of sequences against the average genomic signature of all sequenced reads, then we may be able to distinguish between reads that mapped to the inserted sequence and reads that mapped to the native sequence. So coming back to our plot, to determine which reads map to the Cas9 sequence and which reads map to the rest of the theoretical sequence, we first used a clustering algorithm to identify groups of closely related sequences. Then, for each group of sequences, we calculated their average genomic signature. We also calculated the average genomic signature of all the reads. And finally, we compared the average genomic signature for each group against the average genomic signature of all reads using a statistical test. The tables on the left and right show the results for some of the comparisons that we made. The first column refers to the ID of the average genomic signature of all reads, while the second column refers to the ID of the average genomic signature for each group. The third column contains the p-value result of our comparison, which was calculated from our statistical test. Thus, each row tells us which average genomic signature we are comparing and whether they were significantly different. Specifically, p-values less than 0.05, which are highlighted in green, indicate that the average genomic signature for a given group of reads and the average genomic signature of all the sequence reads were different, which suggests the group of reads were not from the native sequence and hence may have originated from the gene drive. Overall, we found several groups consisting of reads that map to the Cas9 sequence and groups consisting of reads that map to the rest of the theoretical sequence. Besides this, we also found some groups consisting of reads that map to the Cas9 sequence and reads that map to the theoretical sequence. For the groups consisting of reads that map to the Cas9 sequence, we found that some of their average genomic signatures were significantly different to the average genomic signature of all reads. In the same way, for some groups that consist of reads that map to the rest of the theoretical sequence, we found that their average genomic signatures were not significantly different to the average genomic signature of all reads. However, we also found that for some groups consisting of reads that map to the Cas9 sequence, that their average genomic signatures were not significantly different to the average genomic signature of all reads. And similarly, for some groups consisting of reads that map to the rest of the theoretical sequence, we found that their average genomic signatures were significantly different to the average genomic signatures of all reads. So together, our results demonstrate that it is possible to distinguish between some groups based on their average genomic signature, but not others. So why might this be the case? One possible reason is the relatively small number of sequences in each group. And like any statistical test, the relatively small sample size can impact on its reliability. Another reason is because genomic signatures of shorter sequences are noisy in general. This makes identifying groups of closely related sequences based on their genomic signature difficult. So in conclusion, we're developing a method to detect whether something has been inserted into the genome. Our method involves analyzing the characteristic frequency of oligonucleotide sequences, which we refer to as a genomic signature. By analyzing the genomic signatures of sequences, we show that it is possible to detect gene drives that have been inserted into the genome. Importantly, no prior knowledge about the gene drive sequence and no alignment to a reference sequence was required. But besides gene drives, our approach could also be used to detect other types of large insertions, such as viral integration sites and plasmids that contain antibiotic-resistant genes. And whilst our approach is not perfect, we're working towards solving some of the issues that I've highlighted earlier. That being said, we hope that our approach could be applied to poorly characterized organisms. And with that, I'd like to end by thanking everyone in the Transformational Bioinformatics group at CSIRO. And I'd like to thank you for watching this video.